Welcome back, students. For today's class, we will be discussing today on complement system for lecture 8. As discussed in previous classes, one of the important concepts we spoke on opsonization. In that syllabus, in that class, we also discussed about one of the important concept of molecules called opsins, where the pathogen can be attached to opsins, and the antibodies or some other immune cells can go and recognize and kill those cells which are attached to opsins by opsonization. So we already spoke that the opsins may be antibodies itself, or it may be special mannose molecules, sugar molecules. And also we discussed that specifically one molecule called complement C3b, that is one of the protein molecules that also attached to the pathogens that triggers enhanced phagocytosis. So that brings to us to discuss more on the complement system. This is the concluding chapter for Unit 2 and also a very important concept. So the lecture has been divided into the following outline. Firstly, I would like to introduce to the concept of how a damage can happen in terms of the looking in the view of the pathogens. How immune cells kills the pathogen, how they damage them in terms of a mechanistic view or in terms of physical form, physical view. This will be the brief introduction in the first few slides, followed by we will talk more about basics of enzymes in terms of properties of enzymes, different forms of enzyme, active, inactive enzymes, apoenzymes, zymogens, and how enzymes work in a cascade. And one of the fundamental important function of enzymes is proteolysis. Coming on to the other concept of complement system, the present class, we will look more into following questions like what is it, what is the complement system itself, what are the membranes involved in, and then how does it work, what does it do. These are the questions we will look in, try to find answers for these questions in the class, and that constitute the chapters for the syllabus in Unit 2, for example. Complement system components, properties and functions, one of the unit, one of the chapter, and then complement pathways and biological significance. What are the pathways involved in this system? We will talk much about it. Let's take a general example of in terms of looking into an ecological balance, death and decomposition. That's really paramount for ecological balance. In this diagram, you are looking for a specific kind of decomposition of a specific animal that is dead. You look in the slide A, that is called a cadaver, fresh cadaver. Then given a time at a certain time, even day 1, day 2, day 3, day 4, or maybe day 10, day 20, it can be decomposed like this. Let's start from the day 1. You think that all these decomposing microorganisms come and attack in a specific manner and decompose the body of the cadaver in a specific manner into a time, and then comes and leaves the skeletal system only. So it is a systemic process that helps in decomposition. At the same time, you look here, a kind of organized flow of ants coming to drink the honey or imbibe the honey here, given a situation. You see the flow and organization of the ants coming as a specific single unit in a specific three-dimensional circular fashion, completely organizing the sugar here and trying to eat it away, or at certain time the sugar will disappear. In the same manner, the pathogens will be attacked by the immune cells in a specific coordinated manner, specific timeline, where they kill a pathogen given in a form. We will see how they do that. One of the fundamental example here you need to look in terms of decomposition is that the proteolytic enzymes, they play a key role because our body is composed of different membranes and proteins and complexes. The proteolytic enzymes play a key role in decomposing the body here. So now we are moving from the cadaver to moving into the immune cells in terms of how the immune cells physically damage the pathogen, given pathogen. This slide here, this diagram here, you are looking for a body of a pathogen, probably cell membrane of a pathogen, and you are seeing that already it is attacked by immune cells, and the immune system has made a lot of holes, perforations in the body of the animal, body of the pathogen. So eventually the pathogen dies. So here you are looking in one of the complex of the protein, complement proteins, forming a homopolymerization, a polymer complex. That complex go on and attach in the body of the cell membrane, and it can kill it eventually. This is one scenario. You are looking in the physically how they damage the cells. In another scenario, you are looking into a part here where a fresh and healthy living Escherichia coli is here, and it is attacked by immunocomplexes here. And further, this system specifically calls blebbing of the bacteria. At certain time, it explodes, blebs out. Basically, the bacteria kills whatever the above it attached to the immune system, and whatever the immune system in terms of antibody phagocytes, they also complement with another protein complex called complement system. They are also present here, try to kill the pathogen. So coming on to in terms of the basic understanding of bacteria, basic understanding of how enzymes work. Okay, coming back to fundamentals of the enzymes. Enzymes can exist in a kind of inactive form, zymozymes or proenzymes. They can be in the form of active enzyme. How the activation happens, why the inactive form. For example, if there is a substrate here, the substrate has been blocked by specific fragment, polypeptide fragment here. The substrate and the active site of the enzyme cannot access each other. The enzyme, the process, enzymatic reaction cannot happen. Okay, one thing. Then it becomes that some proteolytic cleavage happens, enzymes are removed, substrate attaches, enzyme progresses. So why this mechanism of active inactive enzymes exist in the system? Because the enzymes have multiple functions in the body, and if they have kept active always, they might gonna attack to the substrate whatever they are there. Basically, then the homeostasis will be in imbalance in the system. Basically, it kills its own substrate of the cell that are utilized for some energy reduction, energy production, or catabolism or anabolism. To not to do a thing at a time, what it should not do, the enzymes are put it in a proactive proenzyme process, that's in active state. Okay, the system. Okay, we spoke about fundamentals of enzymes. And in terms of the proteolysis, one of the important concepts, you look into that. Protein in naive condition, different three-dimensional conformation. A protease is the enzyme that cleaves it. It makes nicks. It makes into a smaller fragments. At the end of the proteolysis reaction, it makes into a small peptide. Again, remember that the protein three-dimensional conformation in antigen antibodies, there are a lot of disulfide bonds, peptide bonds are there. A naive state to catalytically active, it can become inactive state. Again, it can come back into active state. Remember that a protein can exist in native catalytic state, inactive state. Again, it can resume the folding and become active state again. So a protein cleaved by some portion can again regain their activity to do one more function of it in a given next second or next time of the reaction. Okay, the screen moving fine? Hello?
Yes,
sir, it's fine.
Okay.
It's moving fine.
Yeah, fine, thanks. And cytolysis we've spoken about here, and also different classes of enzymes, whatever they do, transferase function of the enzyme there, lyase and hydrolase enzymes. So one more important thing, if you understand this cascade, it will be very easy for us to look into the complement pathways. For example, an enzymatic cascade is a sequence of successive activation reaction involving enzymes at a different step, which are characterized by series of amplification stemming from the initial stimulus. Okay? Enzymatic cascade starts with a stimulus and then goes into multiple cascade reactions. At each reaction, multiple enzymes are involved, different enzymes are involved. The enzymes are again, one fragment of the enzyme formed in the previous reaction may be part of the second reaction. It can form a complex with other enzyme. The initial stimulus proportionally goes up into that up to the completion of the enzyme reaction, whatever required at the end product. You are looking for polysaccharide breaking here. It's a kind of in terms of clotting system, but you don't need to look into more details. For example, liposaccharide, factors or enzymes are called sometimes factors. They are involved. Factor C, a process of stimulus, activated C. This activated C goes and involves in the factor B, triggers due to activated B, and then as cascade forms, the pro-clotting enzyme into clotting enzyme, clotting happens. So remember that activated C can trigger another substrate, different substrate, and it make them active. So again, the blebbing, we looked in cytolysis, how this happens here. And for example, here, if you look, for example, this is a complement protein system. you look into the zoom and go into certain methodology, look into the closure of the pathogen, you can easily see that the complement system proteins are coated here. And again, in bacteria also, you can find them, the coating happen here. So coming on to how the complement attaches to the system, how many molecules of the complement are there? You see here, one pathogen, several holes are there in a given surface area. How does it happen? So the abbreviation, or you need to go into a little bit more about definition of the system. The complement system is also known as complement cascade. Is a part of the immune system that enhances. We will see what is the enhancing here. It complements the immune system, or mainly the ability of antibodies and other phagocytic cells to clear microbes and damaged cells from an organism and promote inflammation and attack the pathogen. Basically, a complement system tries to remove the pathogen by using a process of inflammation or attacking on the cell membrane perforation. So it damages basically, right? A complement system damages the pathogen to die. How it does? It does through a combination of immune system antibodies here, and also works through a specific proteins called complements. And basically why they are called complements? Because they are enhancing the function of either antibodies or phagocytic cells, just like opsins we spoken. They are taste buds, they are like the desert molecules. They complement the immune system to do their function in a better way. So why we call them complements? Again, you know from fundamental understanding from previous class that antibody consists of antigen binding site FAB, and it also has a constant region FC and FAB. And we've seen that pathogen recognition and complementarity epitope recognition was done by antibody binding site only. And we have seen that the other domain, the constant region, is important in biological activity, where it triggers and binds to FC receptor to the effector cells. It triggers a specific cascade. And one more thing you have to remember here, that why we call it as complements. If you can imagine a scenario, antibody recognizes the pathogen, it has these protein molecules here. It can become a sniper. It can sniper and it can snipe it out the pathogen like an automatic machine gun, right? I attached it, I know it is a pathogen, I recognize, let's start killing it by like shoot it. It can be dying, that pathogen, right? It can be dying, but it doesn't do like that. Antibody cannot work on its own. It cannot kill the pathogen on its own. That's why it needs a system called complements or specific protein, where the cascades that will come and kill the pathogen. Pathogen lysis and it dies one away. Other process, what we are saying, this is kind of indirect mechanism. A pathogen is recognized by antibody and specific complement system is enhanced in an indirect way. In the other system, in other pathways of the complement system, the pathogen doesn't need to involve antibody. Based on the molecules present in the pathogen, it can directly go and touch in with phagocytes or some other mast cells. They can also kill them. That is direct pathway that we will see. Try to understand why the complement is here. And this is complement system. And what is a complement or complement proteins? A complement is a system of plasma proteins that can be activated directly by pathogen without antibody involvement, or indirectly by pathogen bound antibody, leading to a cascade of reactions that occurs on the surface of the pathogen, and basically it kills the pathogen, it affects our function. That's the definition of the complement. The parts of the complement system, what are the different proteins involved in that? It's a set of plasma proteins mainly produced in the liver, and they are present in the blood, lymph, and extracellular fluids. If you take a fundamental, what is the system at the fundamental level constitute? They constitute specific proteins, plasma proteins, C1 to C9. So C1, C2, C3, C4, C5, C6, C7, C9, they may differ by in terms of their molecular weight. They may differ in terms of their genes located on the chromosome, which gene codes for what. When it codes, they may have differences that may not be necessary for the present class. And one more important thing you need to remember, that they are termed C1 to C9 based on their discovery, ascending order. So initially, let's say in 1980s, C1 was discovered. By 2000 sometime, C9 was discovered. But the mechanism what we are seeing in the complement pathways, no one understood clearly from the starting C1. They don't know how the system works. They know that something happens with complement system. Pathogens are dying. They don't know about the proteins involved in that. They started working it slowly, identifying one protein at a time. So based on the discovery, this was the protein first discovered. Its function basically looked upon, then C5 at certain time, then C9 in the last. So it doesn't mean that in a given a cascade of the complement pathway, they come from reaction 1, C1, they go to reaction 9, C9, that every step is followed by alphabetically to the complement here. It doesn't work like that. It's only an implementation of the discovery when was discovered is that. It may be that these proteins work in a random order in a given reaction cascade pathway, that we will look into that. So a little bit more textual matter, that if you are given an exam, you can write this text and look into that, or make your own notes. It's system where it is present. When stimulated by one or several triggers, the complement protease system, the system cleaves specific proteins into cytokines. The release proteins are cleaved by proteases, cytokines are released, and then not only that, one protease do cleaving one enzyme, cytokines are released in parallel, there will be amplifying cascade of further cleavages. It goes into an exponential manner because in a given shortest possible time, you need to kill the pathogen. Otherwise, the antigens make the body, the cells, organism to die. So a cascade will come up. How it comes, you look into that. The complement proteins are above over 30 proteins and protein fragments, not only proteins. One protein is divided into smaller and larger fragments. Small fragment can do some function, large fragment can do other function. They make up the complement system. Overall, 10% of the globular fraction of the blood constitute the complement proteins. So how the cascade happens, you look into that. Protein can be divided into small and larger, active and inactive. Again, it goes and takes another substrate, it goes into that. One more important things of the complement proteins are that these are heat labile, not destroyed by heating up to 56 degrees. You can make them inactive, and this is also used for fundamentally the physical properties can be used for using a complement fixation test that we will see in further. This is the one more fundamental aspect where you can kill the complements, you can leave the antibodies. Actually, antibodies are heat stable. They can survive 56 degrees without any problem. We will see that in further classes, but importantly, remember that that is an important function. So coming on to pathways, how many pathways are there in the complement system involving C1 to C9 only? No other proteins are involved, or their fragments only. One pathway is called classical pathway, another pathway is called alternative pathway, and the third one MB lectin pathway, MBL pathway. The first pathway, classical pathway, it works through a classical system of antigen-antibody complex recognition. Then it activates a specific trigger of the complement proteins, and at the end moment, they will kill by recruiting inflammatory cells, they kill it that. Coming on to alternative pathway, they also do the same thing, but they don't involve antibody. They have pathogen surfaces they use. They again trigger a specific complement cascade that may be different from this molecule pathways involved here that we will see. An MBL lectin pathway, it's also involves some of the lectin binding on the pathogen surfaces, but the cascade involved in this pathway is almost similar to classical pathway, but this pathway has a different cascade, we will see. But at the end, they go into one of these three steps and they kill the pathogen. Based on the involvement of antibody, this classical pathway is called indirect pathway. And then these pathways, two pathways are called antibody independent or direct pathways, direct association with the pathogen. So, okay, let's probably take some 30 seconds time out and you just look into the different steps here, different names here, different reactions here, what you can deduce. Try to look what looks like common. For example, the blue looks common to me always, not from this point. Just take 20 seconds off, look into this diagram, and then I will come back and resume. Three pathways we are seeing here. Okay, So what I am seeing, whatever I have spoken previously, 3 pathways. This is the MBL pathway, mannose binding lectin pathway, classical alternate. What I do see, whatever is in the yellow, they constitute the part of yellow or yellowish orange. They constitute part of the complement system, complement protein C1 to 9, whatever we have spoken. And these blue ones represent factors or enzymes involved in the system. Enzymes that catalyze specific enzymatic reactions. In alternative pathway, the enzymes are called factors, factor D, factor H. But in other pathways, the complements are named by C1 to C4. And look in the branch here, all these 3 pathways converge on one common enzyme called C3 convertase. And C3 convertase is called by name because its substrate is C3. It divides the C3 into small fragment and big fragment called C3A and C3B. And C3A will have some other small functions we'll see. But the C3B larger fragment again can combine with C3 and it becomes one more part C3 convertase. Later it becomes C5 convertase enzyme. Combining this portion of C3, this small fragment, C5 convertase enzyme will come. And again it will have a different substrate, it will have a different function, different complex. And at the end, everything, all these 3 pathways commonly come into a lytic pathway that is called membrane attack complex, MAC, where it destroys the pathogen here. So look into different components of the brown complement system, proteins involved, look into different enzymes involved. And the enzymes in from here, they have common functions, C3 convertase, C5 convertase, and one is membrane attack complex that destroys the pathway. So one more distinction between the pathways is that from this step upwards, you have different components of the complement. For example, classical pathway involves C4, C1, C2, and mannose pathway C4, C2 alone, and another protein complexes called MASP1, MASP2. And here this involves a factor D, and that quickly jumps from C3B, and almost similarity. We will see what are the systems independently. So all converge upon a lytic pathway that has a specific enzymatic complex. What is that machinery? We call it as the lytic pathway membrane attack complex is formed by C5b, C6, C7, C8 and a multiple multimer of homopolymer of C9 protein. So membrane attack complex C5b, C6, C7, C8 and there is a 9 molecules or multiples of that C9 are there and their function is to perforate, make holes in the cells and make the lysis of that. Okay, remember that. One more important definition you need to remember here: complement activation, complement fixation. Complement activation is nothing but some certain proteins go in a reactive manner. They make this cascade efficient enough so that an exponential cascades can happen. Complement activation is an efficient means to coat antigens so that they adhere to peripheral blood cells and are phagocytous. One thing, but efficiently produce multiple amounts of the complements or complement proteins or protein fragments. Complement fixation is a process of binding of complement to immune complexes or to certain foreign surfaces and make them phagocytic. They are fixed, they are activated, and they are fixed. So this is a diagram that shows Epstein-Barr virus. The virus is here, and without, it's in a native form without attacking to any complement or any antibodies. It's just native form of the virus. The structure looks like that. And this diagram shows the virus molecules, Epstein-Barr virus molecule. They are recognized by antibodies or attached by antibodies here. You see the volume peripheral increases. But from this step to this step, there is no involvement, right? Nothing happened to the cell. They look like native conformation, native form only. Unless the antibodies constituted by complement system here in the C, complement system and antibody together, then you see that complete destruction, lysis of the cell is happening from its form to here. So lysis happens and then the cell dies. So that you can physically in vivo, you can see into that. So how in terms of the C5 convertase, how the enzyme works. This is the end of the lytic step. So you remember, we will go steps from end to the front so that you can understand it better. The ending step is the lytic pathway where C5, one of the complement protein is there. So activation of COI by C5 convertase, an enzyme that involved is called C5 convertase. it leads to the generation of C5A and C5B. C5A is a very small fragment of the C5 complement protein. And then C5B is a larger fragment. The C5B larger, as we spoken, the MAC complex, the C5B attaches to C6C78, C9 multimer. It makes the complex that we discussed, membrane attack complex that makes up here, and then it destroys by cytolysis or apoptosis and cytokine production. The C5A, on the other hand, it will have some other function that it works efficiently again. It goes and binds to some other receptors on cell. The alternate functions for C5A is that neutrophil mobilization, histamine release, increased vascular and tissue factor production. So both of the fragments are C5 is involved in the one of the function, but one is in the complement pathway important for the MAC, others can do parallel function. So coming on to the end, in terms of the complement pathways, how they work in the end product, how they release some cytokines and factors, specifically kill pathogens. We have already discussed several of the fundamental processes. I will just summarize one more time. One thing, lysis, opsonization, activation of inflammatory response, and one more functionality of clearance of immune complexes also. This complement system does that function. First thing is that binding, complementing with the system with antibodies, killing the target cell by lysis. The opsonization, specific molecules and the bacteria, you recognize the opsins, phagocytosis will happen. This, again, a complement receptor, we already spoken in the third class, if I presume. Inflammatory response of the innate immune system, and specific molecules in the blood and plasma, extravasation process comes, and they go and specifically trigger some of the mast cells. Then they go into inflammatory response, kill the pathogen. Clearance of the complexes is again antigen-antibody complexes. Based on the recognition of the attached to the complements attached to this system, they can easily recognize and they can clear certain amount of the immune complexes in the need is arises. So this is summary in terms of the function. Taking into the pathway, how the pathway converges, the components here involved in each pathway, common enzyme C3, C5 convertase, MAC complex will come. But remember that C5A, C5B, C5A, some other function we discussed. Similarly, C4A, C3A, C5A coming from these small fragments of this reaction, they have some other functions. And C3D coming in the alternative pathway, it has one more important function in the B cell activation, saying that the complement system can be crucial for innate immune system also, innate or humoral and cell mediated immune system. One on the opsonization. So coming back again, what we will do now, we will take simply each pathway at a time, try to understand in a better way what are the steps involved in that. Again, if you make this chart on your notes, personally, you practice this chart, I think you will be in a good way to write this pathway analysis here. Classical pathway. So the first involvement are C1, C4 and C2, involvement of C3 convertase. Remember that C3 convertase is a combination of some of the fragments done in the previous steps. It is denoted by C4b and C2a, coming from C2 fragment here, C4 fragment here, large and small fragments. This is termed as C3 convertase. If a bar represent here, that means that enzyme is active. Enzyme is active, it triggers the substrate, attaches to the C3, it triggers another cascade, enzyme step. Then it makes another fragment, C3b, that attaches to these previous two complexes here, B and A, that forms the C5 convertase enzyme. So you need to remember to write down these names, exactly which pathway is coming from what, and then what is the C3 convertase in a classic pathway, what is it in alternative pathway, then you can easily write the cascade. How this recognition happens, the first response is from the coming from complement protein C1, where you are seeing a cell surface, antibodies are here. First recognition happens with the C1 molecule that recognizes antibodies, and then there will be a kind of this C1 has again three parts. What we will see here, C1 is a kind of multi-complex again. It will have a stack protein called C1q, another protein complexes in the polymer C1r, C1s, and these two are tetramers again. They form complex, the stack will form. They have a three-dimensional conformation that looks like this. It recognizes antibodies. It makes the facility of the antibodies and other antigen molecules to come and recognize easily the complex. The steps involved in this here, again, C1q binds the antibody, the complex will form initiation, and then this proper recognition here, the complements make C4 and C2 to undergo enzymatic reaction, a small fragment, big fragment, and then a complex will come, C4AB, convertase will form. The convertase again attacks a lot of C3 molecules, release a specific fragment, then C5 convertase will come up, these combinations. And remember that this is an exponential step. This step will go several times at a time, and it goes multiple exponential ways. That's so many amounts of C5 convertase, C3 convertase can be formed, and then they will go and attack into a form complex. Then C5 convertase attacks to C5, and later they cleaves, and then C5b comes, and then it interacts with specific step-by-step process with the remaining complement proteins. They form membrane attack. You write down specific steps here. If you given a notes on classical pathway, you go in with these steps. And lectin pathway is no much different from the classical pathway, except it doesn't involve the C1. It doesn't involve an antibody, so it doesn't involve C1. So it directly recognizes specific surfaces on the antibodies, lectin carbohydrate surfaces, and this mannose binding lectin go and binds with that. It associates with specific protein called MSAP1 and 2. And then they will help to cleave the first reaction of the complement here is C4, not C1. C1 is not involved. C4 comes, it cleaves, but from C4 to C5 convertase, common pathway in the classical pathway. C3 convertase, same designation, same notation. C5 convertase, same notation. Again, further goes into classical lytic pathway. MFC complex form will come. But in terms of alternative pathway, there is a distinction. It doesn't involve C1 or C4 in the beginning step. It involves a specific chemical process called proteolysis, and it starts with directly forming C3 convertase from C3 molecule by a process called hydrolysis, where the hydrolysis results from C3 to C3A and C3B, and then the C3B hydrolyzed product forms into a convertase enzyme here. Then there is a factor called here. There is no enzyme, factor D here, factor B here. The factors will come up here and then go into a specific cascade and C5 convertase. It is a different designation here. You need to write down and memorize this step. And then once C5 convertase comes here, different step, it takes into lytic pathway in the same way. So how it happens again, you can go into this chart. C3 directly, C3B and C3A, factor hydrolysis. Convertase will come, and one more important here is that the C3B BB complex C3 convertase is not stable in this system, so it needs a properdine enzyme or factor again attached to make this complex stable compared to the other pathways. Likewise, this is one more hydrolysis reaction where C3 catalyzes. This is the C3 molecule. C3 convertase means C3 is divided into C3A and C3B. You look into the bonds, how it looks the C3B activated form. It can go and bind to the membrane, or it can go again trigger form C5 convertase. So membrane attack complex, again, same constitution, common origin for the pathway. Functions are here. And then one more, this you can see clearing immune complexes. The summary